can predict more adverse effects on the clinical drugs or the compound you are interested in. Okay, so I'm going to the brief introduction of our eurofins discovery services. So we have a different sites in all over the world. So I'm present from Taiwan and also in the US, France, UK, and Spain. We have a different sites with different capabilities. So in Taipei side, we are focusing on in vitro off-target profiling, in vitro therapeutic based profiling, and we have some biophysical analysis like SPR-based binding, and also we have some in vivo DMPK study and animal disease models. So this is a brief capability introduction for Taiwan site. So we have like more than 450 biochemical assays, meaning we have more than 400 targets we are studying, and also we have more than 200 functional assays with agonist mode and antagonist mode. So you can see the targets with our targets including like GPCR, ionic channels, NHR transporter protease, phosphodiesterase, MMP, PPT, etc. And also we study on like protein-protein interaction. We can measure the kinetics, bindings, uptake of the proteins and related compounds. And also we do some signaling assays, like we can have some biomarkers, or we study on cell cycles, apoptosis, and also we have some targets for oncology or immuno-oncology, like MLR, ADCC related biomarkers, and also on tumor cell proliferation, migration, invention, angiogenesis, etc. And we have a different kind of technical platform. So we have for the readout, we have a radioisotope radiometry, absorbance, fluorescence TR rate, fluorescence polarization, luminescence, the platform. And also we have other platform like image-based high content confocal image, flow cytometry, qPCR, Luminex, immunoblotting, etc. And we have also very well-established cell culture system and cell bank. And from last year, we introduced a new technology called SPR. So we can also do some biochemical and biophysical assays. And for different therapeutic area, we have like a NASH panel, CNS study, or like FC panel receptor binding assays. So in different kind of therapeutic area, also we are involved in this kind of specific assays. Okay, so today I think our topic is focused on the secondary pharmacology, or we call it safety pharmacology. So here you can see in the left side, we have our compounds or the compound related metabolites. So at the first beginning, of course, we decide our compound to hit a certain target, our desirable target. Either it's a membrane receptor, either it's ionic channel, or either it's an enzyme. So we hope we can see some hits and to target our M target. But sometimes during the drug design, the development, we find something like secondary off-target interactions. So the drug, maybe they bind to our primary target, but also they have some affinities to the secondary target. So this target is not what we want, but they do have some effect, like a physiological effect or even side effects. So this thing we need to really be careful and clarify what happened there. So because this kind of undesirable secondary off targets could maybe produce some toxical effects or some adverse effect, for example, like necrosis, apoptosis, or damage our cell membrane, or some cytotoxicity. So this kind of secondary pharmacology will very highly possibly influence our decision making during our drug discovery. So we need to assess the risk as early as we can and to prevent the costly failure in the later stage. So of course we want to know what happened in the beginning. If this doesn't work, maybe we can change our targets, or we can modify, like here I say, we can reprioritize or remodify our chemical series or modify our chemical structures to avoid the secondary binding. And also this information is very useful to interpret our observed functional responses. In vivo stage, like we have a different observation like behavior change or toxicology, safety changes, or in the later stage with some clinical observation, we can also interpret what happened there. So have to provide a cue, what is the phenomenon behind as we observed. So this is a safety pharmacology guideline. So yeah, always we follow the guideline by FDA. So the categories we have like primary disease therapeutics targets. So we validate by regular biochemical functional study or some phenotypic assays. And secondary pharmacology, we call it off-target profiling. So we can do some in vitro molecular target studies. And we have a lot of assays in the markets like safety screening. So it's very well known by all over the world. We do the screening for all the targets. And also another famous one is HERG. So all the drugs we need to pass the HERG test to know there's no cardiovascular risk. And the other one is the safety pharmacology category. So it's mostly like we perform the safety pharmacology and toxicology in the late phase, like in vivo studies or some GLP-like studies. So this is just the guidelines. And there's a literature doing a survey to the whole pharmacological the industry, how they practice their secondary pharmacology studies. So we can see very rare of the industry, they use their own internal resources. Most of them, they mix their internal resources and combine with outsourced CROs. And a lot of people, they only use CRO. So in the world right now, currently, it's very difficult. We use our own resource inside the company or inside our industry or in our group to verify the secondary pharmacology. But we outsource and we leverage from outside CRO companies. Or we use the CRO companies to combine with our internal data and to make the whole data more completed and interpret more details. So from the drug discovery, like hit to lead optimization or candidate selection, or either later we go to GLP studies. All clients or in pharma, they use CRO to perform the pharmacological profiling for them. Okay, so this is the safety pharmacology profiling panels. So it's our own service products. So I can just make a short introduction. So you can see for drug discovery, we have a hit to lead optimization, and candidate selection. So during this period, the whole process, we need to also identify the hazards or eliminate the hazards and understanding the underlying mechanisms. And so we have here a tier 1, tier 2, tier 3 different categories. So tier 1 is more like narrow. So we have a safety screen 44. And from the US side, we have a safety scan 47. It's a functional assay. So at the tier 1, we can just screening some like potential harmful or potential adverse targets. And if we want to go, it's a little bit more advanced. In tier 2 safety panels, we have safety screening 87. So it's a more advanced. We include more potential off targets inside this panel. And if we really want to know the whole detail, we want to do a more broader screening. So we have tier 3 safety panels. Here is spectrum screen panel or a bioprint profile panel. So it's covered more than 100 different targets, and you can have a more general view of what is your secondary pharmacology and what your compounds really hits on certain type of receptors or some proteins. So according to the experimental design, according to the purpose, we provide from tier 1 to tier 3 different range of safety screening panels. Okay, so here I just like to share our internal case study for 2 different clinical compounds. One is Loperamide, one is Pramipexole. So these 2 compounds already well used in clinical practices. And we use these 2 compounds to see if we can screen something and to find something more by our safety screening panels. So here I can show you this case study and how we design the study and how we apply our technology into drug discovery and interpretation. So here we pick up 3 different panels from our company. So the first one is adverse reaction enzyme plus. So this panel is composed all by the enzymatic assays. So we have a different enzymatic targets. And the second one is our safetyscreen 44. So this is a very typical one, and most of our clients, they start from this safety 44 panel. So they include a lot of GPCR, some enzymatic targets, and some receptors or ionic channels, and or some transporters. And the third one is more broadened. So we have a safetyscreen 87. So it's included more targets in addition to the safety 44. So we can treat it like an expansion of safety 44. So in total, we have total 139 adverse effect reaction associated targets. So we test 2 clinical drugs with CV safety alerts. So you can see all our targets, they cover different therapeutic areas like CNS, CV, endocrine, GI, immunity, oncology, pulmonary, or the others. So in our experiment, we define our hit as a 50% inhibition on our target and at a 10 micromolar concentration. If we find something more than 50%, and we treat it as a hit, and then we do further follow-up studies by functional cell-based assays. So these 2 drugs, they already been reported for some safety alerts in cardiovascular. So Loperamide, this drug is approved in 1976. So used for anti-diarrhea. And the primary target is mu receptor, opioid mu, delta, and kappa receptor as an agonist. And in 2016, by FDA, this drug is reported to have a serious heart problems if we abuse or misuse it. And some cases during 50 years, 48 cases of serious heart problem are already reported. And another drug, Pramipexole. So this approved by 1997. So it is used for Parkinson's disease. Originally, it's to target on primary targets dopamine receptor, D2, D3, and D4 as a receptor agonist. But in 2012, by FDA also, it's an alert that they have a risk of heart failure. So we were testing these 2 drugs to see if our safety screening tester can really find something, and if this result can help us to interpret the side effects proposed by FDA. Okay, so this is our safety profiling. So you can see we have 3 panels in total, 139 targets. So we apply this drug as 10 micromolar. And if the results more than 50% inhibition, and we will treat it as a significant result. And then we do furthermore follow-up dose response tests on functional assays. So we can see more detailedly. So during the scanning, we find yes, the Loperamide, indeed they hit the original targets. So delta, kappa, and mu, so that's okay. So meaning our system is fine. The original target is found out. And then later we want to know, okay, if they hit something else we don't like to see, and if there's some additional hits on these 3 panels, yes. So the answer is yes. So we find they hit a lot of different targets. Like we can see the next slide, that will be more easier. Yeah, here. So except the original delta, kappa, and mu receptor. So this is what we want. And also we find some undesirable targets like alpha receptor, beta receptor, hERG channel, sodium channel, calcium channel. So meaning, okay, it's very possibly this drug will target not only for diarrhea use, but also some risk on heart or cardiovascular systems. So furthermore, we do some functional assay. So here I like to emphasize, so for the binding assay, we are using the radioisotope technique. So we can see the binding and the compound compete with our reference compound under certain receptors. So we can see there's a high affinity binding, or our drug is bind to this certain receptor. But for only from the binding result, we cannot see if this compound is an agonist or an antagonist. So that's why we need a follow-up functional assay to know if it's agonist or antagonist, because all of us, we know agonist and antagonist, they work differently, and their result will be totally reversed. So this is very important to identify either our compound binds to receptor, either is access and agonist or antagonist, or even they just bind but there's no effect. So we always need a functional study to do the follow-up. So here in this case study, we apply this loperamide to our functional assay. So our cell-based functional assay, we have both agonist mode and antagonist mode. So by this method, we can separate agonist or antagonist by our different protocol design. So in our follow-up study, we find this loperamide work as a weak agonist. Weak antagonist against alpha-1b, alpha-1d, D3, and calcium channel 1.2. So you can see the result here. So they're really showing some effects on the IC50, and they shows their weak agonists action. So when we link to the targets and indications, we'll know like that. On targets for loperamide, we expect to see mu, kappa, and delta receptor binding and effects. So it's indication for acute and chronic diarrhea. But for the off-target study, we find, okay, we have alpha-1, alpha-2, D3, or calcium channel binding, and as a weak agonist. So these targets, they are related to hypertension or hypertension, bradycardia, tachycardia, heart failure, cardiac arrest, cardiac stress, vascular thrombosis, or cardiac arrhythmia. So indeed, they do hit some targets related to cardiovascular system. So that could interpret why the FDA they alert loperamide they may have a cardiac risk if you abuse or misuse this drug. So our data provide the proof to the cardiovascular risk. Okay, so for the second case, similar, we tried another drug in CNS treating Parkinson's disease, Pramipexole. So this is a drug for Parkinson's disease. So of course, at the beginning, we find this drug indeed hit the dopamine receptor. This is what we want to see. But furthermore, it's also hit other targets we don't want to see. So we can go here. Sorry. Excuse me. So here we can see this Pramipexole hits a dopamine receptor, but also it hit alpha-2b receptor. So this is not we supposed to see, but indeed this drug binds to alpha-2b receptor. So we do the later on functional assay to see if it's really doing something. So we find a weak agonist. This drug is not only bind to dopamine receptor, but it's a weak agonist against alpha-2b. So what alpha-2b works in cardiovascular system? So we can see here the link. So alpha-2b screened out by our safetyscreen 87. So it's related to some cardiovascular indication, including myocardial infarction and heart failure. So that's why this drug beginning is designed to treat Parkinson's disease, but finally we find there's some risk for cardiovascular system. So there's alerts. When you use this drug, you need to pay attention. Maybe there are some CV related side effects. Yeah, so this is the positioning impact of secondary pharmacology we are looking for. So of course we want to see this kind of adverse effects or undesirable binding as early as possible. And also as wider as possible, we need to screen quite a lot of different targets to know, okay, our drug is really clean, or our drug hit other target we don't expect to see. And then we have to do some more functional assay follow-up to do the dose response curve to know the potency and affinity of this drug. Because sometimes even you hit different target, you have a different binding affinity under different concentration. So maybe in low concentration, you have your medical indication is a good thing you want to see, but in higher concentration, like you abuse it or you overdose, maybe you will have adverse effects. So by collecting all this data, we can do some interpretation on our data and our observation after the drug discovery in different stages. So, but I just like to mention, not only we see always harmful or adverse side effects, sometimes the secondary pharmacology could bring out some potential benefits. So we know inside our body, different receptors or different proteins, they have a synergistic effect. So for example, for the first case, we find the Loperamide is an agonist. At the beginning, we think maybe it's an agonist of H1 receptor. But after that, we know, okay, also the path is affected by this drug. So they have they put together, they have more effects on anti-inflammatory. So this is one good thing. Maybe one drug they target on different targets, but they work synergistically and bring out more potent physiological outcomes. And also we can also interpret why the drug is the drug selectivity and why the drug is so special or unique. So we all know that for the PDE is very highly distributed in our body and also especially in the eyes or in the cardiovascular systems. So like tadalafil, this drug, they are inhibitor of PDE5 and PDE6, but they have a different selectivity. So they are more potent in PDE5. And for the other drug, maybe they are more potent in PDE6. So that will help us to explain why they have this sensitivity. They have the selectivity and how they can work differently on different treatment purpose, or they can really recruit some side effects. And also another thing sometimes makes scientists very exciting because the off-target, maybe they are new targets, and we can repurpose the drug or reposition the drug to the other therapeutic area. So sometimes we decide, okay, we have a drug there for example, for schizophrenia, for CNS, but by chance or accidentally, we find, okay, it's off-target to the other receptor, and maybe they relate to other therapeutic area, like to cardiovascular or to GI, and then we can repurpose this drug from the original use, like from CNS to GI, and then you can reuse this drug or remarket it with different therapeutic indication. So that's also sometimes a good thing we would like to know if our drug have some secondary effects, and if we can use this secondary effect as a future target or future cue to reuse or to repurpose this drug. Yeah, so in the end, I just like to show the value for in vitro pharmacological profiling. So we have a hit to lead optimization, or later on we need to understand the mechanism of actions, and we have a safety screen. So these safety screening panels can help us either to search our on targets or to find out our off targets identifications, and to let us better understand our drugs mechanisms or our drugs selectivities, and to better understand and identify our novel drugs. Yeah, so that's our case study and presentation. So please feel free if you have any questions or if anything I can help you. Thank you.